Thank you for attending the poster session today. I am thrilled to be here on behalf of IntelliReefs and Reef Life Foundation to present the initial results of our flagship coral restoration project. We restore and revitalize reefs through innovative, data-driven solutions, addressing coral reef loss and degradation. IntelliReefs has developed a biomimicking nanotech marine substrate called oceanite that can be tailored for site, species, and function, and cast in any shape and size up to kilometers, allowing for the protection and restoration of entire degraded coastlines. IntelliReefs partnered with the Nature Foundation St. Martin to restore coral reef sites in their MPA following Hurricane Irma in 2017. Large storm surge caused physical damage to the reefs, and intense rainfall increased freshwater and nutrient input. Like many other Caribbean reefs, St. Martin's have also suffered from overfishing, erosion, and pollution over the past 5 decades. We deployed 3 IntelliReefs artificial reef structures off the coast of Phillipsburg in November 2018 to test the performance and biological applications of oceanite in a marine environment for the first time. Our research had 3 main objectives: to quantify and compare the sessile benthic communities growing on IntelliReefs, to determine fish behavior and abundance on IntelliReefs, and to create a preliminary species ID catalog for organisms growing on Caribbean oceanite substrates. The oceanite substrate family is composed of inorganic binder matrices that include pozzolanic components, high-grade calcium limestone, and aragonite. Oceanite substrates were designed to enable precision control down to the nanoscale of surface pH, chemistry, and texture to attract target marine species and enhance growth of calcareous organisms. Oceanite can also be chemically and structurally customized for particular artificial reef objectives. The substrates have an extended pore network for enhancing colonizable surface area, and substrate microtopography can be designed to facilitate coral recruitment and survival. We measured community colonization of benthic taxonomic groups and concurrent fish observations on 3 IntelliReefs in Phillipsburg after 14 months of deployment. 1 artificial reef was deployed at a depth of 13 meters in an MPA that limits boat traffic. A control artificial reef was deployed at 14 meters in a throughway outside of the MPA. The 3rd artificial reef was deployed at 4 meters on a heavily disturbed seagrass bed in Great Bay. To quantify the biotic assemblage on the artificial reefs, divers surveyed each structure in January 2020. All horizontal and vertical surfaces of each artificial reef were sampled with flash photography taken by divers swimming 30 centimeters from the substrate. The adjacent natural reef was sampled using video transects conducted by divers swimming parallel to and at a constant distance from the sea floor. Fish were monitored at 2 of 3 sites using fished cameras on continuous video mode for 10 to 15-minute intervals. Video records were analyzed to identify fish species, frequency of occurrence, behavior, and feeding frequency for individuals greater than 5 centimeters. After 14 months, the artificial reefs were covered in a vibrant community of benthic plants and animals. Figure 3 highlights the differences in the total percent cover of benthic organisms and community composition between substrate orientations and among sites. Horizontal surfaces were dominated by algae and sponges, and vertical surfaces supported a diverse assemblage of suspension or filter feeding invertebrates. The total percent cover of benthic invertebrates was higher on vertical than horizontal substrates for all artificial reefs. We observed the highest total cover on both substrate orientations for the artificial reef in the MPA at nearly 100% cover. Community composition also differed between the artificial reef in the MPA and the adjacent protected natural reef, which is dominated by soft corals. CCA was highest on the artificial reef in the MPA, and both surface orientations had more CCA than the adjacent natural reef. Divers observed macroscopic coral recruits at both artificial reefs deployed in the MPA and in the throughway. This supports the project hypothesis that IntelliReefs can facilitate wild coral settlement in a relatively short amount of time. Based on the number of polyps on recruits, the corals settled during their last reproductive season, less than a year after deployment. We observed that fish abundance and grazing rates were lower in the artificial reef in the MPA than the throughway. Herbivorous and omnivorous fish fed directly off our structures at both sites, indicating that IntelliReefs are food sources for resident fish. The mean grazing rates for fish were 3.89 and 2.66 bites per minute at the MPA and throughway, respectively. This means in the MPA, fish were feeding nearly every 15 seconds. Grazing rates have not been documented on the natural reef in the MPA, and comparisons can't be drawn at this time. We also found that the fish community in the MPA was more diverse than the throughway, but the throughway had an order of magnitude more fish than the structure in the MPA. Increased fish abundance at the throughway may be due to 3 shipwrecks located adjacent to the artificial reef. increasing the carrying capacity of the site. Initial results from this pilot study suggest that when IntelliReefs are deployed in MPAs and low traffic areas, they have the potential to accumulate nearly 100% cover of reef-building species in 14 months. Our research also found that Oceanite's interior matrix was completely colonized by cryptic benthic invertebrates, suggesting that its porous nature can enhance biodiversity. This is currently lacking in all other artificial reef materials. Our results suggest that the design of future IntelliReefs should incorporate large voids for fish and substrates angled to reduce sedimentation and create a variety of light exposure levels to further enhance biodiversity. Our observations of fish abundance among sites suggest that deploying large networks of artificial reefs with high vertical relief over broad spatial scales, we suggest more than 30 meters, may be key to address conservation goals related to increasing the abundance of ecologically and commercially important fish. Evaluating the efficiency of advanced material substrates for enhancing biological and economic objectives on coral reefs is essential for elucidating their large-scale applications. Concrete, rubber tires, steel, and 3D printed artificial reefs do not accommodate the physiological needs of marine organisms. Our upcoming research focuses on the application of IntelliReefs for species-specific recruitment, growth rates of reef-building calcifying species, disease mitigation, and preceding artificial reef modules. We will also examine the application of IntelliReefs into landscape management plans that focus on connectivity, spillover effects, and migration corridors. IntelliReefs increase the customization of artificial reefs and create a unique opportunity to simultaneously target biological, socioeconomic, and cultural benefits across both public and private sectors, returning profit to coastal communities, empowering local people, and improving the scale of restoration success. Thank you.